Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Don Cornegas, and joining me to introduce today's podcast is a man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the double secret selection committee that selects all the guests who appear on STEM Talk.
Hi, Don. Great to be here.
So today we have our good friend Dr. Dominic D'Agostino returning for his third appearance on STEM Talk. And Dom, as most of our longtime listeners know, is well known for his research into the ketogenic diet and the physiological benefits of nutritional ketosis.
You might think after 2 interviews, Dom wouldn't have much more to say about ketogenic nutrition, but you'd be wrong.
You're so wrong. In today's interview, we talked to Dom about his latest work as well as his extensive research into hyperbaric oxygen. So Dom is an associate professor in the Department of Molecular Pharmacology and Physiology at the University of South Florida, and he specializes in neuroscience, molecular pharmacology, nutrition, and physiology. And Dom also is our colleague and a senior research scientist at IHMC. Before we get to our interview with Dom, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review was written by Andy. It reads, "After discovering your show, STEM Talk, I have to say that I was utterly impressed by the outstanding quality of content you provide. The episode featuring Dr. Barbara Thorne discussing termite biology was especially enlightening and left me craving for more." More termites? No. Anyway, nobody in Florida wants more termites. And it left me craving for more. I appreciate your podcast's unwavering dedication to showcasing the most captivating and persuasive thought leaders in the field of STEM.
Yeah, that was a fun interview, and thank you, Andy. And thanks to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to today's interview with Dr. Dominic D'Agostino.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Don Cornegas, and joining us today is Dom D'Agostino. Dom, welcome to the podcast.
Hi, Don. It is great to be back. Thank you.
Absolutely great to have you back. And also joining us is Ken Ford.
Hello, Don, and hello, Dom.
Hey, Ken.
So, Dom, last night you gave a great lecture at IHMC, and there's a full house to hear your presentation. And we'll post the lecture in the show notes for this interview. But one of the slides in particular had a photo of Meryl Streep from her 1997 movie, First Do No Harm. And that movie is about a boy. It's a fictional movie, by the way, but based on a true story. And the boy is 4 years old when he is first diagnosed with severe epilepsy. And despite treatment after treatment and numerous drugs, his health radically declined and his seizures became worse. So the boy's mother, who is the fictional character, as we mentioned, Lori Reimmuller, does her own research, which leads her to Dr. John Freeman at Johns Hopkins Children's Center in Baltimore. And he has a track record of successfully treating children with a ketogenic diet that reduces their seizures. So, Dom, can you give listeners some background on John Freeman and also elaborate on the story a little bit more?
Yeah, glad to do that because John Freeman and the Johns Hopkins Hospital and the ketogenic diet team there were really inspired me to go down the path that led to a research program on ketosis. And that movie, which I watched a decade later, First Do No Harm, I think I watched it in 2008, also really inspired me. So the late John Freeman, he passed away in 2014. He was the director of the pediatric epilepsy center at Johns Hopkins Hospital. I've talked to numerous people. He was describe him as being very compassionate. He taught medical ethics for severe pediatric populations, and he was a prolific mentor. And he mentored Dr. Eric Kossoff, who co-authored books with him. But John revived the ketogenic diet. So he's sort of like single-handedly Jim Abrams. He connected with Jim Abrams to, develop the Charlie Foundation. And the movie, First Do No Harm, was about Jim's son, Charlie, who responded remarkably well to the ketogenic diet. And they think, it literally saved his life. But it was largely a result of John Freeman taking on that case and really saving Charlie's life. He is, described by many people as a fearless and aggressive advocate of the ketogenic diet, even in the face of a lot of criticisms from his colleagues. So he continued to just promote it, and he impacted tens of thousands of people for that. And major inspiration for me.
Mm. And Dom, there was another slide in your presentation that sparked a lot of interest in the audience last night, and it highlighted the numerous evidence-based applications that researchers are finding for the ketogenic diet in terms of treating a wide range of disorders. And we'll include the slide in our show notes, but could you quickly run through these many different applications for the ketogenic diet?
Yeah, I mean, we don't have time to go through all of them, but, it was, I think it's Thomas Seyfried and then another, maybe Dr. Jung Roh, who also inspired me to go down this path. They had mentioned to me early on that the ketogenic diet was likely better for treating other disorders, even outside of epilepsy, or equally effective. And that was largely due to the fact that the diet profoundly changes brain energy metabolism, but also the neuropharmacology of the brain, and really helps to restore the brain's homeostasis. So, I have kind of a bias that the disorders that would be most responsive, and I think the literature supports this, are neurometabolic disorders, inborn errors of metabolism. And that's like glucose transporter deficiency, pyruvate dehydrogenase. But other disorders are pathophysiologically linked to impaired glucose metabolism or glucose hypometabolism. And that could be pretty much any neurodegenerative disease that we talked about, Alzheimer's disease, Parkinson's, even ALS, various disorders that lead to dementia, and all those orders. And also, interestingly, genetic disorders like Angelman syndrome, which we study. So even in the presence of a persistent molecular pathology, a disorder like Angelman syndrome would be responsive to the ketogenic diet. And that's very interesting to me early on, that you could symptomatically manage a disease process. Glucose transporter deficiency is kind of the hallmark, disease that when you implement a ketogenic diet, it can manage the disorder, but you're not changing the genetics, but you're symptomatically managing it. And then there's psychiatric disorders too. There's, schizophrenia, bipolar, and this is a very active area of research right now. So I could go on, but that's just a few of them.
Very interesting. I understand that there's another interesting story regarding a man with chronic obstructive pulmonary disease, otherwise known as COPD, who reached out to you. And as most of our listeners know, COPD is a debilitating inflammatory respiratory condition that is generally progressive and considered incurable. It's my understanding that this 54-year-old man said to you, "I'm going to die, but I think this ketogenic stuff could help me." So, Dom, what happened next? What's the story here?
Yeah, it was something like that. It was an early email, 2016, and his name was Russell Winwood. And I think it was like 2016, he had reached out to me, and he had seen the paper on how the endogenous beta-hydroxybutyrate metabolite could suppress the NLRP3 inflammasome. And he was very well read on the subject of his own disorder, chronic obstructive pulmonary disease, which is really like a death sentence. I did my PhD in a pulmonary critical care department, and there's no good outcomes in COPD. And he took it upon himself to implement a ketogenic diet upon reaching out to a number of people in the ketogenic field, including, later on, connected with Brianna Stubbs and also Nick Norwitz, who is now a MD PhD student at Harvard. And Nick really kind of took this project and made a case report out of it. So huge credits to Nick, who's a super sharp medical doctor in training right now, and also reached out to Dr. Peter Barnes, who's an icon in COPD. And he was inspired by Russell's response to therapeutic ketosis. In 2017, his cytokines and inflammatory markers were off the charts. And this drives the COPD process. And upon initiating and sustaining therapeutic ketosis, those markers went profoundly down. And that had a big response, presumably on increasing his functional expiratory volume and his lung capacity and ability to run. So Russell goes by the title COPD athlete on Instagram or Twitter. So follow him if you can do that. And he posts some inspirational things, including, being able to run marathons with COPD. So he's really an outlier in many ways. And it's just another emerging application that we can add to the growing list of things that are responsive to therapeutic ketosis.
You noted in the case report that there were some limitations that should be thought about. Could you talk briefly about those?
Yeah, well, Russell was a very severe case of COPD. His FEV1 was under 1. And normal is 3.5 to 4.5 liters, I believe. But it was amazing that he could even run, right? But I think it's a synergistic combination of things with Russell is that he's incredibly smart, intelligent. He's very, very optimistic. No doubt that your optimism plays a role in your therapeutic response to this because you follow through with all the little details, including measuring daily ketones and glucose levels. And he also exercises. His exercise probably augments the therapeutic effect of the ketogenic diet. And I talk about this quite often. I touched upon it in my lecture last night is, exercise is a very powerful metabolic therapy. And when it's coupled with dietary therapy, for example, like a high protein ketogenic diet, that could really augment the exercise response. So it's a combination of these things resulted in him being able to reverse a disease process that is universally accepted as something that should not be reversible. So for him to gain lung function and also profoundly suppress those inflammatory markers that were driving the disease process is just truly remarkable. And I think that's why Professor Peter Barnes at New College London, I think he's just, he literally wrote the book on COPD and he was inspired enough to be the senior author on this case report. And I'm hoping that the pulmonary field will take notice and start, spearhead a larger project on this.
Did he engage only in the diet or did he also use exogenous ketones?
Yeah, well, that's something that we kind of skirted around in the case report, just due to reasons associated with conflicts of interest. And also, Russell consumed exogenous ketones at different points of time in his therapy. And when he did consume them, he experienced increased, ability to breathe. And this was very remarkable. And he reported that to me early on. But when we wrote it up, it wasn't, for various reasons, we did not include mention of exogenous ketones in it, but it was part of it. But he really thought that the ketogenic diet was the main driver. Mm-hmm. But I think it does open up the possibility of a ketogenic diet supplemented with exogenous ketones.
Yeah, that makes a lot of sense. And the ketogenic diet, as you mentioned earlier, has been shown broadly to have potential as an anti-inflammatory diet. And since COPD is in many ways an inflammatory disease, your case report, coupled with other evidence, suggests that the ketogenic diet has strong therapeutic potential, not just for COPD, but for other inflammatory disorders as well. Are there other specific inflammatory disorders that spring to mind in this way?
Yeah, it was the publication on the NLRP3 inflammasome, which sets off an inflammatory cascade, including IL-1 beta, IL-8, and, Russell had levels that were off the charts on that. But in theory, autoimmune disorders and many disease, especially chronic disease processes, including Alzheimer's disease, are driven by inflammatory process. If you have systemic inflammation or markers of that, you undoubtedly have neuroinflammation, right? Because the things that uncouple the tight junctions, for example, in the gut too, and the gut is kind of central to this. Those tight junctions are in the blood-brain barrier. So inflammatory molecules like cytokines, IL-1 beta, IL-6, IL-8, these things loosen the tight junctions in the gut, but also in the blood-brain barrier. And blood-brain barrier permeability and endothelial function, it's all tied together and intimately linked to the level of inflammation. And a ketogenic diet, through multiple mechanisms likely, can help to suppress inflammation. And I think it does it very well when there's a driver of inflammation. There's something in the body. It could be an infection. It could be exercise. Various things alter inflammation. And the ketone body, beta-hydroxybutyrate, is thought to be a main driver of that. So it has implications for a wide range of things.
So, Dom, speaking of research, you've been quite busy since your last on STEM Talk, and you've either authored or collaborated on close to 40 papers. And much of your more recent research is focused on ketosis and brain energy. And one of your papers that attracted a lot of attention appeared in Frontiers in Neuroscience. And in the study, you investigated whether therapeutic ketosis by consumption of ketone esters could represent a viable way to treat epilepsy and also other seizure disorders. So can you walk us through this study and also its significance?
Yeah, sure. That study it was more of a review of different studies, and it was kind of a bold approach to propose this idea of ketone administration for seizure disorders. And it was an overview of the history, the rationale, and the use of ketone esters, which are probably the more powerful ketogenic agents as metabolic therapies. And this paper was encouraged by a mentor of mine that helped me get into this field, Dr. Jung Ro. And we collaborated on this. And the key points of the entire review of the literature was that cellular metabolism profoundly modulates neuronal excitability, right? And a ketogenic intervention, whether that be a ketogenic diet or ketone supplementation, alters our physiology in ways that change not only brain energy metabolism, but the neuropharmacology of the brain. And now this is widely accepted. It was more of a theory about 10 years ago. And then the ketone bodies, beta-hydroxybutyrate and acetoacetate. Acetoacetate seems to be uniquely favorable in this regard. Exert anti-seizure effects in animal models and in various neurometabolic disorders. And the level of acetoacetate seems to correlate really well with seizure control. So the general sort of overview of the literature strongly suggests, and this was sort of the consensus too with neurologists at American Epilepsy Society when we talk about this, that therapeutic ketosis represents a clinically viable treatment for epilepsy and other seizure disorders, not just the ketogenic diet, but the field needs to recognize and acknowledge, and funding agencies need to support alternative approaches to inducing therapeutic ketosis and managing that in patients. Many patients just are unwilling or unable to successfully implement a ketogenic diet for various reasons. So that was really the central point of that review and the various studies in it.
For listeners who are not familiar with ketone esters, they may want to check out episode number 54 of STEM Talk and our interview with Dr. Brianna Stubbs. Dom, could you give our listeners a short primer on ketone esters and discuss why so many researchers are really pretty excited about their potential?
Yeah, I'd love to do that. I'd like to give kind of a shout out to Dr. Brianna Stubbs. I mean, she was a pioneer in the field of ketones and performance and just understanding ketone ester metabolism in general. And I know her well, and her scientific and athletic background really fuels her passion. She's kind of a living experiment in progress when it comes to that regard, a scientist and an athlete. So the ketone esters have the greatest potential to rapidly elevate and maintain high levels of ketosis. And they're very important from a clinical perspective because you're unable to achieve 3, 4, 5 millimolar concentration of ketones with MCT oil, for example, or ketone salts. If they're formulated correctly, you can get up to 2, maybe 2.5, but not 3 and 5 millimolar of ketones. So in that regard, ketone esters really do have an advantage. But I like to say, as with glucose, higher is not always better when it comes to levels of ketosis. It certainly is for some of the things that we study, like oxygen, central nervous system oxygen toxicity or glucose transporter. You do want higher ketones. Then, of course, you can also take just a lower dose of a ketone ester. You don't have to shoot up to 5 millimolar. So the level of ketosis needs to be appreciated and taken into context, because if you elevate your ketone levels high, which only ketone esters do have that capacity to do, that metabolic burden is the kidneys need to deal with that, and it stimulates ketonuria. Also, when you elevate ketones above 2 millimolar, and I've done a lot of insulin measurements to validate this, and I'm in communication with people who have also done this. If you have a delta, so if you're on a ketogenic diet and say you're like at a level of 2, and then you take ketone esters and shoot to 4 or 5, or if you have no ketones in your body and you shoot up to 2, and you go above that, that releases insulin. And if various ketone formulations, if you take that and you're below 2, there's a very small, if any, insulin response. So something about a 2 millimolar delta seems to release insulin. And what this does, it could potentially decrease endogenous ketone production, which is tightly regulated to the suppression of insulin signaling. So that needs to be taken into consideration if you're to use these ketogenic agents, that you could suppress your own ketone production if you bolus it in a way that elevates your ketones rapidly over 2 millimolar. And this is kind of a little pet project of mine, and we've published it, or we've talked and presented it in abstract form, but we plan to do a paper on this.
So Dom, we've talked a little bit about ketone salts, and we've talked a lot on STEM Talk about ketone esters. And there's another category of product out now, principally from a company, Kinetics. Have you looked at that, and how would you describe that formulation?
Yeah, my understanding is that Kinetics is a combination of R13-butanediol, which we are very familiar with, and we've published, more than a decade ago on animal models, 1,3-butanediol. It's a combination of 1,3-butanediol and beta-hydroxybutyrate salt. So, and that's a clever combination. Some things to consider, though. So the ketone ester, a ketone beta-hydroxybutyrate monoester, is 1,3-butanediol with an ester bond to beta-hydroxybutyrate. And that could be subject to degradation. And then you kind of end up with 1,3-butanediol. And I remember asking the late Richard Veach, when I was looking at his patents and stuff and thinking about various formulations that we wanted to use in the lab, and I asked him, well, why don't I just use 1,3-butanediol? I was trying to acquire the monoester for some experiments in our lab. And he was like, well, 1,3-butanediol will make you drunk and it needs to be combined. And it will give you a buzz and but it will also elevate ketones. It's a dose dependent thing. When you combine 1,3-butanediol with a ketone salt, then you get that extra kick in ketones. And it's a pretty unique formulation. And we have tinkered with these things in the lab. I would like to mention that if you take 1,3-butanediol over a period of time and measure your liver enzymes, your liver needs to metabolize 1,3-butanediol to make it into beta-hydroxybutyrate. And we've seen this in animal models and I've seen it in blood work, this will elevate AST and ALT enzymes over time. So keep in mind that 1,3-butanediol, a limitation is the mild narcotic effect that it has. Some people like that. I think that could potentially reduce its ability to enhance cognition because you start to get buzzed, but you wouldn't want to take 1,3-butanediol and try to elevate your ketones in the 3 millimolar range because then you start to get some mild narcotic effects that some people like. But so, it's a unique formulation and I think it has some various applications, but I think it's going to be limited. In particular, the use of this in the elderly population that may be a little bit unstable because when you take enough of it, you do feel a little bit dizzy. So you just proceed cautiously with it and also maybe do some blood work to look at liver enzyme function.
Those are all good points. And nobody that's tried it here has felt any narcotic effect, but they've only been having a single dose. They haven't been.
I think they've dosed it in a way to prevent that problematic effect that could affect them as a company.
Sure.
Yeah, but we have 20-liter containers of 1,3-butanediol, and I've experimented with it quite a lot. And it has a remarkable effect at lowering blood glucose. And our experiments are really dialed in on that. It's a hypoglycemic agent. And it's doing it because it takes a lot of energy for the liver to maintain gluconeogenesis. So when you take 1,3-butanediol, it causes a redox shift in the liver, and the liver has to work hard to detoxify it because you do get an aldehyde that comes out, and that's toxic. And then that reduction in liver energetics decreases gluconeogenesis. So that's kind of why glucose goes down.
Yes, there's another company, not this company, another one actually selling it as hard ketones. In other words, as like a drink like at a bar,
Yes.
It's an amazing world.
Less toxic than alcohol. So if you want a buzz, maybe that would be favorable. But at the same time, you are producing a toxic aldehyde that could, is potentially toxic and your body has to clear that. That does not happen with ketone salts. It does not happen with MCT. So I'm of the opinion that a well-formulated electrolyte balanced ketone salt with MCT is a very safe and viable option if you want to enter the exogenous ketone space.
So, Dom, you've had a number of studies published since 2019 looking at ketone-induced neuroprotection, which is an area of research that our group is definitely interested in. So can you give us an overview of some of this work?
Yeah. So a lot of our research, focused on oxygen toxicity, and there are many different ways in which therapeutic ketosis can have a neuroprotective effect for oxygen toxicity. Oxygen toxicity alters redox signaling, neuronal excitability, and cellular metabolism. And some early work with ketones was showing that in a cell culture preparation or even in a brain slice preparation, the ketones could preserve cellular bioenergetics, calcium signaling, and suppress oxygen-free radicals in a way that would suggest they're enhancing mitochondrial function. So the neuroprotective effects of ketones seem to be really a part of enhancing that mitochondrial function. And then by preserving mitochondrial function in the context of a stressor, and this could be glutamate, it could be, a variety of different toxins like in the environment, oxidative stress, then that was the main driver. And it's kind of going upstream instead of throwing on antioxidants, which maybe you remember, Dawn, when you were actually doing your PhD at the time, a lot of my focus was on antioxidant compounds, and we still do focus on them. But if you go upstream and target mitochondrial function and bioenergetics, then you are preventing oxygen-free radicals just by enhancing the mitochondria, right? So you're kind of working upstream. And I think that's the real sort of neuroprotective benefit of that. And that applies pathophysiologically to pretty much every neurodegenerative disease you can think about, right? If you go upstream and give the mitochondria a form of energy that produces proportionally less oxygen-free radicals and also more ATP, then by virtue of maintaining the bioenergetics of the cell in the context of environmental stressors, that can apply to so many different disorders. And that's what I saw under the microscope in cell culture and then hippocampal cells. And then we moved to animal models. And now, as you know, we're doing some research at Duke in humans.
Yeah, and just as a side note to the listeners, CNS oxygen toxicity is specifically of concern for either hyperbaric oxygen therapy or divers who are breathing elevated pressure of oxygen during dives. And Dom and I actually met. I just realized that was 15 years ago.
Wow.
15 years ago.
Time really has flown.
I know. I was doing my PhD research and he was doing his postdoc research for the Office of Naval Research at the time in the undersea medicine community. So it's amazing to see how far this work has come and all the different applications. So speaking of that, one of the animal studies in which you participated looked at Angelman syndrome, which you talked about a little bit already, but just to kind of go back to this, it's a rare genetic and neurological disorder that causes seizures, developmental delay, loss of body movements, and lack of speech. And in this study, Dom, you explored whether ketone ester supplementation could mimic the ketogenic diet as an anticonvulsant. And you also examined the effects of ketone esters on behavioral and metabolic outcomes in your mouse model. And this was an interesting study because it presented strong evidence that ketone ester supplementation offers a promising mitigation strategy for many of the devastating aspects of Angelman syndrome. So could you talk to us about some of the key takeaways from this study?
Yeah, sure. I'd like to acknowledge Dr. Stephanie Charloni, and she was a PhD student who worked on this project. Dr. Ed Weber, who was in my department, is the leading researcher on, preclinical and now clinical therapies for Angelman syndrome. And the title of that paper, which was the Angelman syndrome mouse model, was Ketone Ester Supplementation Attenuates Seizure Activity, Improves Behavior, and Hippocampal Synaptic Plasticity in that animal model, which is a very good animal model. Not all animal models are good. That's a very good animal model. So there was a number of things that were discovered in that animal model research in regards to mechanistic, how it was working, and behavior. The ketone ester was not gavaged to the animal, it was integrated into the diet. So we took a standard diet and then we integrated the exogenous ketone into that diet, and then the animals fed on that. And it more or less suppressed the phenotypic things that were associated with Angelman syndrome with that animal model. And the learning behavior, and that was probably the main thing, right? Learning behavior was improved dramatically in the hippocampus. There was enhanced synaptic plasticity. And mechanistically, one of the things that I was really excited about, that it validated something that an idea or a theory that I had, was that therapeutic ketosis with a diet or with any form of supplementation increases the GABA to glutamate ratio. And in that particular study, they measured glutamic acid decarboxylase 65 and 67. And it restored GAD 65 and 67 were elevated. So you had a greater conversion of glutamate, which is hyperexcitable and potentially toxic, to the brain stabilizing GABA. And as you know, Dawn, Claude Pianadosi and other people have looking at these enzymes as sort of things linked to CNS oxygen toxicity. So it ties into some of that oxygen toxicity work, this glutamate to GABA ratio. And I also think things like adenosine 1 receptor signaling, and another area of research that we're focusing on is the ability of ketogenic therapies to elevate NAD levels. So NAD levels go up quite a bit, and some of our new metabolomic data further validates that. So it's steering me into the direction to look at NAD precursors and to study that in oxygen toxicity and then compare that to, there are a number of influential people in the field that feel like this NAD is really driving mechanistically, some of the therapeutic effects of ketosis.
Yes, it's easy to see how the diet would elevate the NAD pool. You wouldn't take as much out of the pool, essentially.
Yes.
But with exogenous ketones, do you see the same thing?
Yeah, so there is good evidence for that, and I don't want to talk too much about it because I'm so excited about publishing it, but the evidence would suggest that you're modifying TCA cycle intermediates in a way that's generating NAD and the reduced intermediates that drive the electron transport chain and NAD levels. The metabolomic experts that are reviewing, analyzing some of our data told me without me even asking them that this suggests the NAD pool was increasing significantly, in various tissues and not just brain. So we're looking at muscle, we're looking at, a variety of different organ systems too.
Yeah, I could see how that was the case with the diet. I wasn't sure if that would be the case with the exogenous.
Yep.
That's interesting. In the past decade, there have been numerous human studies that have looked at the therapeutic role of ketogenic diets in various neurological disorders. And recently, there's been quite a bit of research examining the potential of ketosis to help with Alzheimer's. Can you touch on some of this research? You addressed it a little bit last night in your lecture here at IHMC. And before we even get into it, our listeners might want to check out episode 59, where Stephen Cunnane talked a good bit about Alzheimer's in the context of exogenous ketones.
Yeah, Stephen Cunnane is doing a lot of remarkable work, and I was pointed in his direction by Dr. Mary Newport, who actually gave an IHMC lecture in 2013, I think, and kind of have to give credit to Mary and her husband, Steve, that they got put on my radar in 2008 when a St. Pete Times article came out. And I was just getting into this field and applying it to the oxygen toxicity project. And actually, the first grant that we got on ketosis was from the Alzheimer's Association through Dave Morgan, who was the CEO at the time. And there was a preclinical study on mouse models of Alzheimer's, there's some caveats to using those mouse models. And then a clinical study, too, on MCT and coconut oil, which kind of flopped at the U.S. Alzheimer's Center just because people didn't want to take the placebo. If it had any potential for helping them, they just wanted to go to the nutrition store and get MCT oil. So there's a wide range of reasons why ketosis could be therapeutic or beneficial for Alzheimer's disease. And Cunnane's work is really remarkable in that he did a variety of scans, a dual PET scan, looking at cerebral metabolic rate with glucose and also with ketones, specifically acetoacetate, and found that as we age, our capacity to metabolize glucose for energy decreases gradually, whereas that same decrease is not observed with ketones. And the implications of that are that ketones could essentially fill the energetic gap if there's glucose hypometabolism, which is more or less universally accepted. Alzheimer's patients are not always told to have an FDG PET scan, but if they do, it shows due to atrophy of the brain and also probably vascular deficiencies and blood flow deficiencies, but there's a decrease in glucose metabolism. But it's also linked to a decrease in the GLUT3 transporter, pyruvate dehydrogenase complex deficiency, and also the activity of that enzyme is decreased. So it's multifactorial. But the important thing is that ketones kind of circumvent many of the pathways that would normally block glucose metabolism. So I think there's a big potential there. And I think there's a subset of Alzheimer's patients who would respond. Remarkably interesting, Steve was ApoE44. And the literature as it is right now suggests that would be not as responsive. And I observed with my own eyes his response to a fairly significant dose of MCT coconut oil combination. I watched tremor stop and I watched him become more animated. And that was 2009. And that increased my enthusiasm for going in the direction of Alzheimer's, which I was a little bit hesitant. I was more confident that it would work for seizures, exogenous ketones. But I think there's a lot of potential there. And I think government agencies should be funding this research because there's not a whole lot of things that are working.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
And Dom, you were part of two studies that came out back to back earlier this year that looked at the effects of a ketogenic diet on athletic performance. So high carbohydrate, low fat diets have for decades been the predominant nutrition strategy for athletic performance. And recent evidence has challenged that superiority of carb loading in terms of athletic performance. So in February this year, our former IHMC colleague, Dr. Andrew Kutnick, published the results of a study that had middle-aged athletes undergo two 31-day isocaloric diets. So a high-carb, low-fat diet or a ketogenic diet and a randomized counterbalance and crossover design while controlling calories and training load. And you, as well as Jeff Bullock, participated in the study. And we're wondering, what were your key takeaways from this study?
Yeah, thanks for bringing. That's a relatively new study that came out, and it was done in the lab at Grove College by Dr. Philip Prinz and his amazing team of researchers. And former IHMC employee, Dr. Andrew Kutnick, my former PhD student, really took the reins on this study and was first author on it. And the main takeaways were that a high-carb, low-fat diet pitted that against the low-carb, high-fat diet in this crossover design. The main takeaways that over 4 weeks of adhering to this diet in a particular group of people that were competitive athletes between the ages of 30 and 50, I think male athletes, that the high carb, or going to rephrase it, the low carb, high fat diet, when it compared against the high carb, low fat diet, which is basically the conventional approach or diet used for athletic performance, there was similar short duration performance with high intensity, even upwards of 85% of VO2 max. So this is a bit unheard of. So it kind of broke new grounds as a study in that regard. And we matched differences in calories and training load, and the body composition alterations were also similar between the two diets. And also glucose reduction is often inversely correlated with fat oxidation rates. So that was a major take home of the study, that as glucose consumption or carbohydrate consumption correlated with blood levels of glucose, and those blood levels of glucose, as they went down, that was highly correlated with fat oxidation rates. So if you want to become fat adapted and a high fat and ketone oxidizer, looking at glucose with the CGM, and props to Levels Health, they sponsored part of it with their CGM devices and software. So that was one of the big takeaways, that there was really no performance differences and that there was significant differences in glycemia between the groups. And that was pretty remarkable.
You and Jeff Volek collaborated with Andrew Kutnick on another study that came out in March that looked at what in exercise science is known as the crossover effect. You mentioned this earlier. First, before we discuss this study, though, could you explain for the listeners what exactly is the crossover effect?
Yeah, so this crossover effect paper was really just kind of a review of this theory of the crossover effect. So the existing data did not really incorporate low-carb, high-fat diet. So it kind of challenges this idea. I guess the simplest way to put it is that fat oxidation is directly proportional to the intensity of the exercise. And when the intensity of the exercise gets above 85% of VO2 max, the existing paradigm of the crossover point would extrapolate and predict that fat oxidation would essentially be zero when your VO2 max is over 85%. That you would just be, highly glycolytic, almost completely glycolytic. So the data from our study that we just talked about challenges that and is kind of rewriting the crossover effect is quite different in the context of a low carb, high fat diet. And the fat max is reached during the exercise intensity. With the crossover point, it's generally around or predicted to be 0.3 to 0.6 grams per minute. And we know that in the context of being fat and keto adapted, that it's double that. And that fat oxidation rates are significantly elevated even in the context of going above your VO2 max. So that was sort of the take-home message, because the dogma is that high-carb, low-fat diets produce superior results in exercise performance by increasing carbohydrate and glucose availability. That you need that availability, and that's absolutely essential for high-intensity exercise. But your body has incredible metabolic flexibility. Your body will metabolize what you eat. And I think that's a really important concept of the incredible capacity for metabolic flexibility, which is observed in shifting this crossover effect.
Thank you. It's a clear description. Appreciate it.
So, Dom, you've been part of many studies that have demonstrated the positive impacts of a ketogenic diet, but let's talk about the elephant in the room, and this has come up in other podcasts as well. There are lean individuals with low triglycerides and high HDL cholesterol that sometimes develop a marked elevation of LDL cholesterol when consuming a very low carbohydrate ketogenic diet. And this phenomenon has been termed the lean mass hyperresponder phenotype. It has been a much discussed topic for several years now, and is something that we often get questions about here at STEM Talk. And you and other researchers had an article last year in the Journal of Clinical Lipidology on this topic. Can you talk about this article, which was titled, by the way, Elevated LDL Cholesterol Levels Among Lean Mass Hyperresponders on Low Carbohydrate Ketogenic Diets Deserve Urgent Clinical Attention and Further Research?
Yeah, this is, as you know, a very hot topic in this field, and debated hotly, too, because of the atherogenic risk associated with elevated LDL, in particular ApoB. So a lean mass hyperresponder, and I think maybe it was Dave Feldman and maybe a few others in his group that coined this term, exhibit extreme increases in LDL on ketogenic diets. And that increase in LDL is directly proportional to the level of carbohydrate restriction. So there's no doubt about that, because when you reintroduce carbohydrates back into the diet in someone who is a lean mass hyperresponders, and I kind of put myself in that camp, then the LDL starts to fall back down to normal. And I can almost get it back to normal if I just get 100 grams of carbs in, which is not a lot. So this is quite reversible. It's also very dynamically reversible in a fairly compressed time frame. So, should this be a concern? And I think the main takeaway of that article, and Dr. Ron Krauss was a senior author on that. And again, major props to Nick Norwitz, soon to be doctor at Harvard. He was kind of motivated us all to write this and coordinated the group. It's kind of like herding cats. But the cardiometabolic risk factors typically associated with lean mass hyperresponders are low. Everything else is low. So you have really high HDL, very low triglycerides, typically, low blood pressure. Everything looks great, on blood work except the LDL, and if you were to measure it, the ApoB in these people. So the idea then is that this needs to be studied. So that was the general consensus and the message of the article, that it really needs urgent attention, and we need to understand this because quite a few people are using very low carb diets to not only manage, epilepsy, which I talk about, but to manage type 2 diabetes, to manage their weight, to manage many different chronic disorders. And you're going to have, I thought the population was 5 or 10%, but it's more like 20 or 30% that have this phenotype. So we need to longitudinally measure things like coronary calcium, do a CT angiogram, measure the intimate thickness, and do that in lean mass hyperresponders versus non-lean mass hyperresponders over a long period of time. So that needs to be done.
Also, LDLP typically is greatly elevated in that group.
Absolutely. Yep.
Yeah, I'm also in that group.
Yeah. And you probably find, well, I don't know, if you were to transition to consuming more carbohydrates, you'd see a reversal of that.
Yeah.
And I guess the question is, why are you following a very low carbohydrate diet just for performance and other risk factors? But I think, could you achieve, I think the field is kind of questioning, could you achieve the same enhancement of metabolic biomarkers or other things that you're looking for with the diet that's low carb, but not very low carb? And will that decrease your atherogenic risk associated with that increase in LDL/ApoB. That remains to be determined, but I know just experientially that when I'm in a state of ketosis, which needs to be with a very low carb diet, unless you're always taking exogenous ketones, that I feel there's a certain level of enhancement and lucidity and energy that I get with being in that very low carb state. And I'm not willing to let that go.
No, I mean, there's no free lunch.
Yes.
And so there's a trade-off on everything. And there
That's right.
are pharmacological means to deal with some of these issues as well.
Yeah. Like a tiny dose of a statin, for example, if you're elevated.
Say that if you're in the category that you're describing, the hyper responder, they tend to not require a large dose of the statin, a tiny dose typically. Often paired with Zetia or something like that. If you're
a hyper absorber, which I've not experimented with that, but I'm going to do a Zetia experiment to see if I am a hyper absorber. I think that I might be.
So, Dom, we often say there's no one-size-fits-all diet, and what works for some people doesn't work for others. And I know some people who breeze through a ketogenic diet and others who don't seem to tolerate the diet as well. And you had a review that appeared a few years ago that examined genetic and other markers in an effort to identify how people might respond to a ketogenic diet. And your goal was to identify individuals who are most likely to benefit from a ketogenic diet and also pinpoint other individuals who may be at increased risk for adverse health outcomes because of the diet. And you've described this as a clinician-friendly review of state-of-the-art research into biomarkers for a ketogenic diet. So can you walk us through this review?
Yeah, sure. So big props to Lucia Aronica, who was the first author, I believe, on that review. And she's really an expert in epigenetics. And as you said, it looked at very common SNPs that could predict our response, beneficial response versus negative response to a ketogenic therapy. And it ranked these SNPs in regard to their strength of the scientific evidence and in regards to outcomes that were not so much focused on neurology or epilepsy, but weight loss, body composition, and cognitive performance. So the ApoE was discussed in that. So there's, different rare mutations could profoundly affect the ketogenic response. And I see this in people that email me that they feel sick when they go on a ketogenic diet. And I suspect that they have a fatty acid oxidation disorder or their body's not producing ketones. And, 10 years ago, I didn't really think about SNPs or just genetic reasons for this. But within this review, it went through a number of different biomarkers that could be looked at. Some of the ones that come to mind are the LIPF minor allele, and that particular coded for a gastric lipase, right? So there was a 17% people had this allele and they produce less gastric lipase and they lost more weight on a ketogenic diet. So that was kind of interesting. And maybe they're digesting less fat. So more fat, less is digested. So it's kind of a calorie wasting thing. And there's a GYS2 allele that had to do with hepatic glycogen. So that was correlated with a better response. And then there was a number, not going to go through all of them, but the ApoE4 too, and it talked about the ApoE4 allele. And as Ken knows, and we talked about this a little bit last night, there's a 3 to 15-fold increase in people who are ApoE4 who are homozygous for that. And that is something to consider with the ketogenic diet. There's a number of different discussion points when I think about Alzheimer's disease or dementia and the genetic risk for that. There's this idea of a boulder going downhill. And I think I saw it in one of Nick Norowitz's talk. It was like this boulder, you can't stop it. And different lifestyle factors can push it down faster, right? But we want to slow the boulder. And there's a number of different contributing factors to that. And I think lifestyle plays a huge role, blood-brain barrier breakdown, endothelial function. But if we look at different populations that are, for example, people in Nigeria, elderly Nigerians, there's no association of ApoE4 with cognitive function or Alzheimer's disease. But if you move those people to United States, then you start to see that resurface. And there's another group, the Italians, the Southern Italians. So Southern Italians who are ApoE4, stated another way, gives them no risk for dementia. But if they move to the U.S., then that becomes reversed. So a main thing is our genes will not necessarily predict, our response to the ketogenic diet. And there are many different reasons for maintaining therapeutic ketosis, even if you do have a genetic risk. For example, being in ketosis reduces NF-kappa B, MMP9, which are drivers of blood-brain barrier breakdown. And these are driving Alzheimer's disease and various things associated with glucose control, right? So when you're in a state of therapeutic ketosis, your glycemic variability goes down pretty profoundly. And glycemic variability is tightly correlated with endothelial function. And endothelial function is really highly correlated with cognitive decline. So there's a variety of different lifestyle factors or metabolic characteristics associated with following low-carb diets that are quite favorable for people who are ApoE4 carriers, in particular homozygous.
Dom, another area of research in which you've been heavily involved centers around hyperbaric oxygen. And this is a well-established treatment for decompression sickness, which is, of course, a potential risk among scuba divers. Hyperbaric oxygen therapy, however, is now being used to treat several medical conditions, including traumatic brain injury. And there is controversy regarding this. Could you give our listeners a short primer on hyperbaric oxygen and why it has lately attracted so much attention?
Yeah, hyperbaric oxygen therapy, a limitation of that is oxygen toxicity seizures. So that is what I actually studied the negative effects of hyperbaric oxygen, and most of my work has been focused on that. But we did observe that, for example, mice live longer with cancer with hyperbaric oxygen. But taking a step back, hyperbaric oxygen therapy, there are 14 FDA-approved applications of hyperbaric oxygen therapy, ranging from carbon monoxide poisoning to decompression sickness to ischemic wounds, radiation necrosis, for example. So hyperbaric oxygen therapy has a really profound effect, and I've seen it on wound healing. And traumatic brain injury is a type of wound. There's ischemia, there's a decreased brain blood flow, and a decreased oxygenation. And doing hyperbaric oxygen therapy, particularly in the early stages, people who are younger, like in animal models, the animal literature is very clear on this too. It's quite muddy, but it's clear that if you intervene quickly and you have a younger animal, that will be more responsive. So there's a therapeutic response to hyperbaric oxygen therapy and a variety of different mechanisms associated with that. But if you give too much of it, it could potentially be pathological. And with traumatic brain injury, you do have a compromised patient, so you need to consider the dose of hyperbaric oxygen therapy and whether that is pathological if it's too high, because it does cause reactive oxygen species production and potentially oxidative stress. You need to personalize it to the person. And the adaptive response to the hyperbaric oxygen therapy is really what's doing the healing, I think. There's potentially a release of stem cells, a mobilization of stem cells that hone in on sites of injury. Of course, you're reoxygenating the area. So there's a lot of different factors that need to be taken into consideration with delivering hyperbaric oxygen therapy, especially for TBI. But you want to start low and then kind of work up from there. And I'm of the opinion that it shouldn't be done, 5 days, Monday through Friday, but there should be a break, 3 days per week. So every other day, for example, Monday, Wednesday, Friday, with a break in between.
So, Dom, speaking of hyperbaric research, it probably won't be a surprise that I'm going to squeeze in some questions about Nemo. So for our listeners, this is a NASA project that sends crews of astronauts and aquanauts, as well as engineers and scientists, to live on the bottom of the Atlantic Ocean in Aquarius, which is the world's only undersea research station. And Nemo provides a very convincing analog for space exploration by mimicking the high physiological stress environment that astronauts experience during space missions, and that they'll eventually experience on the upcoming Artemis mission to the moon and eventually a human mission to Mars. So I was fortunate enough to be on the crew of Nemo 21, and you were on the crew of Nemo 22. And your wife, Dr. Chilla R.A. D'Agostino, was a Nemo support diver during your mission, and then she became a member of the all-women crew on Nemo 23. And for all three of us, I know that Nemo was such a wonderful experience. And I know that you were primarily selected because of your extraordinary work at the University of South Florida Hyperbaric Biomedical Research Laboratory and your research into how extreme environments impact the human body. So, Dom, can you talk about your experience and also your research with Nemo?
Yeah, I think, Dawn, you can relate that the Nemo experience as a whole, everything from training at NASA in the neutral buoyancy lab to just being among the NASA scientists and the astronauts and training with them, living with them, and doing research with them, it ranks as one of the best experiences of my life, really. And I got to thank IHMC for that, too, because I think that evolved out of a Blue Sky mission, and then where I met Bill Todd, and then you got on Nemo. You probably put in a good word for me, and somehow I got on Nemo too. And then Sheila, as you know, impressed people with her mission control skills, so she got on Nemo. So it was amazing. We collected a lot of data. But man, I just can't say, enough good things about, Bill Todd and Mark and the Hyperbaric techs, the life support guys, Otter and Sean, and Shell was my commander. But Nemo is, a mission in the Aquarius habitat, and it's a very unique extreme environment because you're exposed to hyperbaric air, which is high levels of nitrogen and oxygen. And also what really interests me too was the high levels of CO2. Especially when the alarm went off and I was doing some calculations on the parts per million of CO2, it was like 10 to 20 times higher. And when that alarm went off, that kind of, and we quickly had to change. I don't know if you got into that scenario too, we had to change the scrubber out. But all that, it makes it for a very extreme environment that mimics the space station in many ways, especially the CO2. And it's just an amazing opportunity to do research, physiological research on a lot of different variables associated with being in a confined space and being in an extreme environment. So, Dawn, and you did microbiome research, and actually I've been meaning to reach out to you because we have, your mission, my mission, and Chilla's mission, microbiome samples, I think. So we need to, that was just one of the, I think we had 5 or 6 IRB protocols that had to go through IHMC, and I thank IHMC for that, and then NASA and also ESA. So it was quite a mission just to get the IRB protocols for all that research through.
Yeah, it's incredible the amount of research that your team did, and then also just in general. I think for our mission, we had, over 20 something projects going at once. So there's a lot of data collected across the board, not just for human physiology, but also, looking at different types of hardware and software and all types of different things, engineering projects. I know we did a lot of projects with marine biology as well as simulation of research that would be taking place on the planetary surface, for example. So really cool. I agree. And I did not have the CO2 experience, thankfully. So sorry to hear that you guys did.
Well, the astronauts have different takes on that. I know Scott Kelly mentioned, I think I counted 58 times in his book that the CO2 was giving him headaches. And then other people like Shell said that CO2 really did nothing. He felt perfectly fine. But Samantha Cristoforetti, the commander of Nemo 23, was like, she could definitely feel that when the CO2 levels were high, that she would just feel totally off. It was like a breath of fresh air when they restored the function of the CO2 scrubber in that. So that's kind of an interesting scenario. But the astronauts that I talked with, they just said that Nemo is very unique from all other space analogs that I'm aware of, in that it's truly an astronaut training mission. And they all said it's the closest thing, that you can get to sort of being in space and living in space in regards to analogs. So that was pretty cool. And it was just a unique experience to train with them and to do tests with them. And, the top tier level of expertise, with your commander and my commander, and just, being at the center too, and then training on all the engineering projects that are ultimately going to be used on Artemis is just so thrilling that it's almost like a dream that I was part of that. So.
Yeah, I absolutely agree with you on all of the above.
Yep.
So, Tom, you followed up this mission with a paper in 2021 that was titled Human Adaptations to Multi-Day Saturation on NASA Nemo. And you examined the physical and also the psychological burden that aquanauts experience living in a multi-day hyperbaric environment. And you looked at parameters such as heart rate, weight loss, blood flow, cognitive impairment, and mood. So can you walk us through this paper and also the significance of this work?
Yeah, I mean, there's a lot of data jammed in that paper, and thanks again to former IHMC Dr. Andrew Kutnick. He was first author on that paper. We observed, I think the remarkable thing was that this extreme environment did not really produce any profound alterations in these variables. There was a significant drop in body weight. So that's kind of concerning, considering especially it was only like 9 or 10 days and I lost almost 10 pounds, I think. And I was eating a lot. So the EVAs take a lot out of you. And sometimes you're real busy. You just forget to eat, and maybe you drop some water weight too. And sleep was quite profoundly altered. My total sleep time, I think, went down, and this is the crew as a whole, about almost an hour to 40. But my deep sleep, so we used the Oura Ring. We also used the Polar V800 chest strap to look at sleep. The deep sleep was, I was always over 2 hours of deep sleep every night. I was just getting 1 hour total, less sleep. But I felt refreshed and I felt fine. Temperature went down, heart rate went down. And the general data was pointing towards an increase in parasympathetic activity and a decrease in sympathetic activity, which is very interesting considering the context of being immensely task loaded during that. But I felt, as soon as I got in that environment, and I wanted to ask you this, Dawn, I felt a sense of calm in that environment. The air is heavier, obviously. It's, 2-3 times more dense almost. And it's also a little bit higher in nitrogen. And I was thinking that you're right on the edge of just the threshold for experiencing, nitrogen is not narcotic, but nitrogen under hyperbaric pressure becomes mildly narcotic. So I was thinking that could contribute to maybe the calmness that I felt. But I think when you're in the flow and everything is organized on, what was that timeline that we had to, what was the name of that software that they use, I think, on the space station? The red line, you got to keep in front of the red line.
Yes.
I found that strangely calming. And it's like, I knew what I had to do. I was like, okay, I'm going to do this in our lab. I'm going to put everybody on the timeline, all the students in the lab. And I think it's stressful to think about, but I think when you become very task loaded, but you know what you have to do, it was kind of calming. So autonomic function, it was just amazing that our bodies could adapt. We've just evolved at living at one atmosphere. And you think you throw someone in, 2 to 3 times the amount of pressure that things would go haywire, but and live for under saturation, but it doesn't. So that's pretty remarkable.
I thought that the sympathetic, parasympathetic, that was really interesting. And I think is exactly what you're saying. I think that you get into this flow state, like you said, and it's simplicity, right? Like you just know what you're there to do and you're doing it and everybody's in sync. And so, I felt like you're just kind of in this zone. And I think you probably feel that in a lot of different spaces. You probably have athletes that feel that, explorers that feel that in different situations, military personnel. And I've talked to them, when they're in certain settings, it's just like you're doing there what you're trained to do, and that your entire focus is on that part of things. So it just makes things a lot simpler as far as things go. So,
and you're not reacting to stimulus like emails,
social media,
and text messages.
Exactly.
That's a very good
exactly.
point, Ken. Yep.
It just keeps everything very simple, and it's like, this is what I'm here to do.
It's like being a human.
Yeah.
What?
Dawn, how did you sleep? Because we didn't track sleep in your study. So I meant to ask how you slept, and everyone had like a little bit different opinion, but the data supports towards less sleep, but people felt like they were rested. So I slept
very well. Yeah,
I did too.
We slept very well. We didn't measure, so I don't know about deep sleep versus overall sleep time, but I always felt very well rested. The one thing I will say is several of us, it was kind of mixed in our mission, but several of us gained water weight. I know I gained water weight, and I think it was the high salt food because we were eating that camping food, the dehydrated camping food that was reconstituted. So super high in sodium. But I will say, like, about 5 days after the mission was over, I lost all that. And overall, I lost weight, right? But I had put on probably a significant amount of water weight I think it's just because of what we were consuming.
So you came out heavier than when you went down?
Yep.
Wow. Okay.
Yeah.
So
if you see pictures, I was like a little bloated. But in 5 days, like overall, I lost weight, like compared to before we went in for the mission.
Yeah.
So it was all just water gain.
Well,
but I really chalk that up to the food because we're just eating such high sodium content food.
Yeah. And I didn't have that food. I brought my own food. I don't know if you know my situation, but I was determined to stay in ketosis, and my ketones were above my glucose for the whole, like I stayed in high ketosis, and I brought a lot of chicken, MCT oil. I poured over it. They didn't allow me to bring sardines because it was a smell issue. But yeah, I was getting 4,000 like plus calories per day, and I ended up like losing 10 pounds, which is kind of weird.
Wow.
You're in ketosis.
Yes.
Yeah.
Yep. And the EVAs, even though the water is like in the mid to upper 80s, I believe, after like a 5 or 6 hour EVA, and I came out and I was like shaking kind of uncontrollably. My body does dump a lot of heat in that, and I'm kind of sensitive to that, but it took a while. And that's when my ketones were the highest, and my glucose was something like 50s. or 40s or something really low. My
Super
ketones
low.
were like 6.7, which
Wow.
was the highest. It was right after an EVA, and I did some psych measures. So we have a lot of data still that we have not published. We're sitting on like 4 papers. The microbiome, we need to get that out, Dawn, but there's taste, and there's a taste manuscript, but the NASA TLX, the GAD7, SQ, NBAC, PHQ9, like we're sitting on a lot of data we still need to publish, in part with Steve Fiore was part of that, looking at team cognition and things like that. So we're sitting on about 4 or 5 manuscripts that we still got to get out.
That's awesome.
So this has been great, Dom. And but before we let you go, I understand that you and Celia just moved, not just, but a little while back, moved from Tampa to a farm.
Yeah.
Could you tell us about your life on the farm?
Yeah. Wow. And it seems like not too long ago, but it was like 5 years ago, I guess we moved to the farm. And, farming, I think it really keeps me grounded. Actually, literally, yeah, you're working on the farm, and I like to run experiments. So we bought a farm that was a row crop farm, but we phased all that out, and I put in, we have cows, we have sheep. I'm planting different types of seed right now, sun hemp, experimenting with animal feed and also equipment. When I get back, I have a forestry mulcher coming. So we have timber that we grow, and being a farmer, really, you got to be a mechanic, you got to be a vet. A big thing that I'm excited about is we're developing what's called silvopasture. So we have trees that are growing, and ultimately, once I finish the fencing, I'm going to let the animals roam among the timber, and they will maintain all the area. So basically the pasture will grow under the trees, and I'm very excited about that. So farming is fun, and I think it just it hits a sort of an itch that I had needed to scratch, because I grew up farming and I loved it, and I'm kind of nostalgic about it. So I'm really enjoying it and having a lot of fun.
I can imagine it's a total shift from living in a large city and the typical existence.
Yeah. Yep.
Well, Dom, this has been great catching up with you. Thank you so much for joining us again on STEM Talk.
Thank you for having me. I appreciate it. It's been great.
It's been great fun, Dom, and we appreciate you coming on STEM Talk for the 3rd time.
Thank you for the invite, Ken. I appreciate it.
Absolutely.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Well, I've always loved talking to Dom. He's not only a great researcher, but just a truly good person. And I particularly enjoyed our conversation today about hyperbaric oxygen research and, of course, about Nemo.
Dom is really a multifaceted researcher. I thoroughly enjoyed watching the way people responded to him at last night's lecture. We will be sure to provide a video link to the lecture in our show notes for this episode. I encourage listeners to check out the lecture. Dom did a great job at the lecture. And in particular, he sort of highlighted how now we have more than 400 published studies demonstrating evidence-based applications for ketogenic nutrition. And as we discussed today, it is, in fact, an extensive and impressive list.
If you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage, where you can find the show notes for this and other episodes at stemtalk.us. This is Dawn Cornega signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.